Hello, my dear students. Right now, today, you have the last in the schedule lectures on general hygiene in actual semester. As you can see at the slide, this lecture is about food poisoning and its prevention. Well, first of all, traditionally, let me give you the definition. What is food poisoning? As you see, food poisoning is acute, not contagious diseases result of eating food massively contaminated with a certain microorganism or with the toxins. So it can be bacterial or non-bacterial origin and unspecified etiology. And we should consider bacterial or microbial food poisoning, non-bacterial or non-microbial food poisoning, and unknown etiology food poisoning sometimes occurs. As it was mentioned, first of all, what's the main difference between food poisoning and intestinal infection? Because it has the same symptoms, but I should emphasize again, food poisoning is not contagious disease. It means it cannot be transferred from sick persons to healthy. Instead, like in case of infection disease. Usually, bacterial food poisonings characterized with a high number of affected persons, and it has symptoms of short-term infection. Usually, it's caused with some opportunistic, less often pathogenic microorganism, but it may cause severe intoxication. Usually, it's associated with entry to gastrointestinal tract, food massively contaminated with living bacteria or the toxin emitted during process of life and death of bacteria. And there are three main conditions absolutely necessary for foodborne disease occurrence. The first point, bacterial contamination of food. Second one, if food is contaminated and if violation of cooking technology happened, it leads to the next stage, wrong food storage or out-of-date shelf-life food also can cause to food poisoning, like first and second points. So to cause food poisoning, we must face with food bacterial contamination, violation of cooking technology, or wrong food storage. And we have two extra additional factors for foodborne disease occurrence. The first of them is compliance of chemical composition of food product to biochemical and microbiological characteristics of specific pathogens. So specific food may be the good media for specific pathogens. And the second one, when pathogen contaminate foodstuff in sufficient amount is critical mass is necessary, because ready and cooked food not primary sterile. For example, sour cream or cottage cheese or yogurt contain their own microflora, and to contaminate food and to cause food-borne disease, pathogen or opportunistic bacteria, first they need to overcome the natural biological barrier. It's like competition and fight with natural microflora and food products. And when considering bacterial food poisoning, it's necessary to subdivide it into the food toxic infection and food toxicosis. Food toxic infection can be caused with Escherichia coli, with Proteus vulgaris, Clostridium perfringens type A, Bacillus cereus, Streptococcus faecalis, and others. Bacteria, a lot of bacteria, in fact, may cause food toxic infections. Who is the source of food contamination? Usually, source is a living organism, people or animal. What about people? It's maybe sick persons or recovering convalescent or healthy bacilli carriers who sick with enteritis, cholecystitis, appendicitis, paraproctitis, any inflammation process associated with infection. And I should share that the most dangerous source of infection is a sick patient with subclinical forms of diarrhea or respiratory disease, but not healthy bacilli carriers, but convalescent recovery persons who look almost healthy, but still diarrhea or some signs of respiratory disease, because they are maybe not provided with medical checkup. However, in this case, they emit a lot of pathogens. If we compare number of emitted bacteria, it's several times more than in case of bacilli carriers. And animals, also sick animals or animals bacilli carriers, can be source of food contamination. How food may be contaminated with pathogens? For example, it maybe happened with contaminated equipment at food industry, or with contaminated kitchen tool when cooking food at home or at catering. It can be contaminated by rodents, by rats, mice, with dirty hands when cooking food, in case of we don't follow sanitary procedures when cooking food, with contaminated water or with soil particles. What's the main epidemiological patterns of foodborne disease manifestation? First of all, it's very short incubation period. As mentioned, it's about 24 hours after eating contaminated food. Almost simultaneous disease among persons who have eaten the same food products, and widespread of disease in case of centralized sale or of contaminated food. So a lot of persons affected can be united with the same supermarket or the same cafe. And it's called territorial limitations. All of them are in a limited area. And fast termination of morbidity is common when withdrawal from sale contaminated food. When we stop eating, so it leads to fast termination of an outbreak. And persons who hadn't eat contaminated food stay healthy, no disease on them, because it doesn't contagious from sick persons to health. And seasonality is common, usually in our country, for example, in our climate, it's seasonally is typical. It's usually it happens in the warm season. It's in summer. What is the main clinical symptoms, main clinical manifestation of foodborne disease? First of all, stomach and intestine symptoms. It's acute symptoms of intoxication or intoxication in the stomach, because of penetration of pathogen into bloodstream. It causes symptom of intoxication, like with bacteria or with toxins. So we can reveal fever, chills because of bacteremia and toxemia. And general intoxication symptoms like infection, for example, vomiting and nausea, abdominal pain, diarrhea, and as a result of body dehydration, when a affected patient loses too much liquids and electrolytes, it leads to secondary symptoms of poisoning. It can be observed blood circulatory disorders, decreased blood pressure, dizziness, fainting, cold skin, cyanotic skin, especially in distal part of extremis, pale bluish shade of skin of face, especially of nose and lips and ears and hands, feet, and dry mucous membranes. How to prevent foodborne disease? You must know, keep in your mind. The first group measures aimed at the prevention of food contamination and cooked food with pathogens. For example, we need to review among food industry employee who are sick or bacilli carriers with pathogenic or opportunistic bacteria, and we need to treat and sanction them timely. Careful sanitary and veterinary supervision of animals who are the source of food products absolutely necessary, especially if we need to focus for young animals first of all. It's a task for veterinarian service. The next point is to provide a strict good personal hygiene and sanitary regimen when product food in food industry. Absolutely necessary is to avoid contact between ready cooked food, semi-cooked and raw food products. We need to separate them when cook food everywhere, in kitchen or in food industry, catering, in cafe. To transport food, we must use specially designed transport with the special design containers for food products only. And disinfection of kitchen tools and equipment used for cooking food should be provided. We need to fight against insects and rodents in food industry. Second group of measures aim to provide condition when prevent massive multiplication and of microorganisms in food. For example, we need to store food products, cooked food in a cold place in the fridge when temperature not exceed 6 centigrades. And sealed cooked, for example, first and second dish at cafe should be at the temperature above 60 centigrades. And as you can see, the university cafe, for example, first and second dishes are they sell it at the hot condition. At the same time, some salads, cold drink should be stored and sell at 14 grades of Celsius and less. And observance of cooking technology is also absolutely necessary. We need to provide sufficient heat treatment of food to destroy pathogens, foodborne disease, to provide a thermal neutralization of conditionally suitable products if it's used. And we need to maintain strict observance of sale term and shelf life of food. And what temperature should be used? As mentioned in this picture, you can see that the maximum temperature should be used when cooking whole poultry or chicken breast is a big piece of poultry of chicken to kill all the bacteria. What's about food toxicosis or food intoxication? It's acute disease arising due to eating food massively contaminated with toxins produced by specific pathogens. What's the difference between foodborne toxic infection and intoxication? Food intoxication, pathogens don't participate in the pathogenesis of food poisoning. And in this case, intoxication occurs because of exotoxin produced by bacteria. And these exotoxins emitted to the food before eating of contaminated food. And food toxicosis and emission of exotoxins provided with bacteria like Staphylococcus aureus and Clostridium botulinum with some molds producing toxins like Aspergillus, Fusarium, Penicillium, Crevis purpurea, or fecal toxicosis. For example, paralytic shellfish poison and diarrhea shellfish poison. Now let's consider Staphylococcal intoxication. It's occurred more than 80% of cases of food intoxication caused with Staphylococcus aureus enterotoxin. And the most common food products for staphylococcal toxemia are milk and dairy product, meat and meat products, especially cutlets, for example, salads containing eggs, dishes made from poultry, fish, mashed potatoes, some type of pasta, and cakes and confectionery products. What's conditions necessary for Staphylococcus aureus to produce the exotoxin? So the temperature in milk, for example, room temperature during 8 hours, for example, or in cutlets, in cod and mashed potatoes, and semolina porridge, this time is about 3 or 8 hours. And confectionery products in cakes, if sugar concentration less than 50%, toxin can be produced in 4 hours at 37 grade of Celsius. And if we store the food at 4 grades of Celsius, in this case, Staphylococcus aureus toxin doesn't accumulate it in food. And main source of food contamination with Staphylococcus aureus are sick humans and sick animals who are related to milk farm, for example, if milkmaids or food industry enterprises or confectionaries sick with some pustular infections on their hands like pyodermia, panarium, and some burns, or sick patients with angina, pneumonia, otitis, catarrhal disease in upper respiratory tract. And sick animals, if they are used for milk producing, cows, sheep, goats, for example, it can be added to additional source of infection. And in case of mastitis, if they use these animals in farm for milk producing or meat, in case of its contamination, it can cause the Staphylococcus aureus intoxication. What's the main clinical symptoms of Staphylococcal intoxication? Severe acute gastroenteritis. So severe cramping pain in the epigastric area related to nausea, vomiting, and diarrhea. And usually body temperature is normal or subfebrile, It happens because of pathogenesis, intoxication, provided with already produced exotoxin from food, but not because of endotoxin produced by living bacteria after eating food. And severe signs of intoxication like weakness, prostration, muscle cramps, decreased blood pressure, thready pulse, acral cyanosis. Well, how to prevent staphylococcal toxicosis? We need to cook food carefully to prevent food intoxication. And sick persons, especially who are sick and contaminated with staphylococcal aureus, mustn't cook food. They should be avoided before its vaccination and treatment. We need to prevent growth of bacteria in food, maintain proper storage, and veterinary control of animals should be provided. We need to provide timely identification of personnel with any inflammatory disease in upper respiratory tract and posterior skin lesions, and remove them from cooking food before the treatment. And they mustn't contact with already made with cooked dishes. And for example, in our countries, in catering enterprises, we have a special journal, registration journal of health, where working employee who came for work every day, they put their signature every day. They must confirm they're absolutely healthy and they haven't any symptoms. Also, we need to prevent colds among food industry employees and to provide a timely sanation of patients with a teeth infection, sore throat, and provided timely disinfection of kitchen tools and food items. Strict observation of food sanitary hygiene at food industry and personal hygiene absolutely necessary, like strict observation of cooking technologies, proper heat treatment regimens. So it means enough temperature and enough exposure, and especially when provide unconditional temperature storage, condition and timing perishable products. Timely sanitary and veterinary control for health of animals in milk dairy farms and slaughter, and provided safety of raw food products of animal origin. Now let's consider botulism. What's botulism? First of all, the definition: it's severe, maybe the severest food poisoning of bacterial origin associated with eating food contaminated with neurotoxin produced by Clostridium botulinum. Botulism occurs, it's related to eating food of usually homemade conserves. For example, salted and pickled mushrooms in hermetically sealed containers, dried or smoked fish, canned vegetables, fruit, and meat and fish, and homemade ham fillet, sausages. And what's the condition necessary to produce toxin by Clostridium botulinum? Spores germination delayed because of high concentration of sodium chloride, if it's concentration over 8%, or with high concentration of sugar over 55%. And acidic environment if pH less than 4.5 units. But consider, keep in your mind that Clostridium botulinum is anaerobic bacteria and the lack of oxygen is necessary for vegetation from spores to bacteria, and it grows up to 50 degrees of Celsius and in a range of pH between 4.7 to 9 units in anaerobic condition. I should emphasize again, Clostridium botulinum is anaerobic bacteria. And you must know botulinum toxin is highly resistant for stomach acids and proteolytic enzymes. So it doesn't destroy it in the stomach. And you must know that there are 4 main types of botulism. Nutritional, it's a classical, the most common form, infant, wound, and unknown etiology. What's the main clinical signs of botulism? Incubation period lasts in the range between 12 to 36 hours up to 8 days. And it depends on the time of penetration of toxin through lymphatic and blood system to central nervous system, to the irreversible binding to nerve cells and bulbar centers of the brain. Non-specific symptoms, for example, like common weakness, fatigue, headache. Sometimes acute gastroenteritis can be found, like nausea, vomiting, diarrhea, and constipation sometimes occur. But we need to focus on specific symptoms of botulism. Specific means neurologic symptoms of botulism, for example, related to the vision disorders, for example, ophthalmoplegia and double vision, diplopia, weakness of clarity of vision like grid or fog before eyes, ptosis of eyelids, strabismus and anxiety when eye pupil dilatation is unequal in right and left eye, and the lack of reaction of pupils to the light, and nystagmus, paralysis of eyeball. As for pharynx, we can review oranoplegia and another pharynx disorder like voice disorders up to full aphonia and full absence of voice. Swallow disorders when liquid food, liquid drinks came outside through nose. It's called dysphagia or aphagia. So these neurological and ophthalmological symptoms have happened because of paralysis of facial muscles, and it leads to facial emotions are limited or even absent. Salivation are reduced, but body temperature doesn't match pulse rate. Usually it's normal body temperature or even a bit less than normal, and very fast pulse at the same time. Some complications in lung, for example, pneumonia or myocarditis, cystitis, pyelonephritis with a rise temperature, motility disorder in gastrointestinal tract due to paralysis of intestine muscles, and constipation and flatulence happens. And finally, the most dangerous complication is paralysis of respiratory muscles, and death can be caused with a paralysis of respiratory muscles because of stop of breath. And summarizing of botulism symptoms, let's revise again. It's double vision, blurred vision, ptosis of eyelids, voice disorder, swallowing is impaired, dry mouth and lack of salivation, and muscle weakness. And in these pictures, you can see ptosis of eyelids. How to prevent botulism when cook conserves at home? For example, we need to add enough amount of sodium chloride and acid, organic acids, when cooking mushrooms at home. Vegetables and fruit should be washed from soil particles, absolutely, because the soil is a natural environment for Clostridium botulinum spores. And we mustn't use spoiled and damaged vegetables, fruits, and berries for making conserves. And to maintain a high level of acidity when cooking homemade preserves, for example, adding acetic acid. We need to observe all necessary sanitary and technical requirements according to the guide of cooking food when make homemade ham, sausage, fish, and we need to follow term of storage, absolutely. And sterilization when package of food when conserves is necessary too. When cook food in food industry, they need to provide it fast process of raw food and timely evacuation when cooking meat. Widespread use of cooling and refrigerators if deal with raw materials, and it's forbidden to sell conserves with a sign of blood. It's blotting. Now the next group of foodborne and bacterial food poisoning is mycotoxicosis. Mycotoxicosis also is nutritional diseases caused by eating food contaminated with mycotoxins produced by microscopic fungi, produced by molds. For example, you can see example of these molds on the pictures. Aspergillus flavus. Flavus means yellowish. It's like kind of molds producing toxins. Or Aspergillus flavus toxin is really dangerous because it's toxin in case of chronic exposure when eating affected food during one time. Usually corn and wheat is affected. In case of improper storage, it promotes a risk of liver cancer. Penicillium also, it's like molds and produce some toxins. For example, usually penicillium affect damaged and spoiled fruits and vegetables in case of improper storage. Ergotism. Ergotism is severe mycotoxicosis. It happens in medieval and the middle centuries in Europe. It was like severe outbreaks characterized with gangrene and convulsions because Clavis purpurea, when affect corns and wheat, produce composition of lyserginic acids, and it can affect nervous system. It has severe effect on nervous system. It can cause even hallucinations and cramps and gangrene. What's about fecal toxicosis? Fecal toxins produced by one cell algae. Usually it's related to seafood. For example, in the warm season, in ocean, from June to October, mussels, clams, oysters, scallops can be contaminated with toxins produced by one cell dinoflagellate. And these episodes of intensive growth of dinoflagellate leads to a high concentration of their toxins, paralytic shellfish toxins, in water and seawater and ocean water. And it can be absorbed with a sea living organism. And the main effect of this toxin is affection of nervous system, neurotoxin. And consider its resistance for cooking, like Staphylococcus aureus toxin is resistant for boiling. It's just one way to avoid, to prevent food poisoning with paralytic shellfish toxins is to stop catching of seafood in the period of red tide in oceans. What about non-bacterial food poisoning? You should know that there are 4 types of non-bacterial food poisoning: plant origin, animal origin, mushroom poisoning, and chemical poisoning. Let's consider them. First of all, there are a lot of food poisoning with plant origin. Usually it happens with young children and adolescents because of their curiosity. And a lot of wild and domestic plants contain poison substance in their leaves and berries. Let me show you some pictures, some example of poisonous plants. It's, for example, nightshade, castor bean, dumb cane, jacquerie bean. And we must know let's look at the pictures. What's very bright but dangerous plants. For example, hemlock has contained very strong poisons that it can be eaten, added in food and salads by mistake, for example, because it looks like some spices, herbs. And Heliotrope europeum, Lasiocarpinum. Just look at this picture, try to memorize how it looks to avoid eating of these plants by mistake. Sometimes wet and corns wet can be contaminated with wild seeds of poisonous. For example, wet can be contaminated with ryegrass, with zerno, wheat seed. And to avoid food poisoning with this one, we need to provide harvest collection timely before it will be grown. Atropa belladonna. And in pharmacology, they use a drug atropine, and first in the history it was found and produced from Atropa belladonna, and these berries contain too much atropine, and if eaten by mistake, it can cause severe intoxication, especially in children. Nightshade berries, so bright but contain solanine. And ricin, contained in castor beans. It's dangerous harmful toxin affect stomach, heart, nervous system. It can cause cough, weakness, lung disorder, heart failure, fever. But proper use of castor oil helps in industry, in medicine. Tacos bacata, beautiful, bright, but poisonous berries. Just avoid eating of them. Dieffenbachia, it's a common and rather spread domestic plant at the public buildings, living accommodations, and we need to follow our children to avoid, to prevent touching and eating them accidentally. Indian bean also contain hemagglutinin, and as for red kidney bean, to make it safe, we need to cook it in proper way for several hours. And hemagglutinin can cause diarrhea and vomiting. Sawanin, this name related to the name of sun, and because it's accumulated, for example, in potatoes, if it's stored under direct sunlight, and potatoes become green. And also sawanin can be accumulated in green tomatoes, in eggplant, for example. And this alkaloid sawanin should be removed. This green part of potatoes must be removed when pale. Potatoes, and peel and wash, and make potatoes safe. But pay your attention, we must be careful when cook jacket potatoes. Sometimes it can cause poisoning. As for animal origin food products, usually the most common with fish. Some type of fish, especially reef fish, contaminated with ciguatera toxin. Horse eye jacket, king mackerel, yellowish snapper, hawkfish, parrotfish, black grouper, amberjack, dog snapper, giant barracuda can be contaminated with ciguatera. In the most common areas in the world shown in this world map, and the red areas, ciguatera fish poisoning endemic in these areas, and sometimes it occurs in the areas in oceans marked with yellowish color. It can be in Atlantic Ocean, Indian Ocean, in Pacific Ocean. We should memorize what fish species can cause ciguatera food poisoning. And these bacteria dinoflagellate toxins accumulates not only in fish but some another sea animals. Usually it's common for tropical reefs, for tropic seas and tropic parts of ocean. And cooking doesn't destroy ciguatera toxin, and it's non-effective. Just one way to avoid it. Don't catch this fish in case of intensive growth of dinoflagellate to avoid ciguatera fish poisoning. Scombroid fish. Sometimes scombroid fish can be the source of food intoxication too. For example, scomber, marlin, herring, tuna, sardines, mackerel, amberjack, shad. If histamine converted by bacteria into if histidine convert to histamine by bacteria, it can cause intoxication too. And how to prevent histamine poisoning eating by scombroid fish? Its concentration of these biologically active substances of histamine should be checked in the food, in the fish, exactly before it's one more poisonous fish is common for Japan, puffer fish or tetradon poisoning. And this dish is delicious dishes traditionally in Japan. And in Japan, only very specially certificated chefs who had passed special exams have rights to cook this fish. Because in case of improper cooking, it can cause death because of respiratory muscles. And sometimes happens when extremely consumers ask to chef leave some part of poison. Just imagine they ask to chef left some remaining amount of poison in fish to feel some sense of risk and dangers. It's very strange request. And mushrooms. Mushroom poisoning in case of improper collecting poisonous mushrooms to be mentioned later. Let me show you some slides. It's a false blusher, fly agaric, discap, destroying angel. So we must avoid collecting these mushrooms when collecting mushroom in forest in general. False blusher mentioned in the picture. Fly agaric, rather bright and beautiful but poisonous mushrooms. Discap also. Look at this destroying angel. Is one of the most dangerous mushroom in the world. It can affect liver, it can cause death, even just eating of one mushroom per family, in case it cook like champignons, and in case of added accidentally, it can cause death of all family. How to prevent mushroom food poisoning? Everybody must collect only well-known mushrooms, and children and adolescents must be guided by adults when collecting mushrooms. It's absolutely forbidden to sell mixed mushrooms. Only sorted by species mushrooms can be sold in the market. And homemade foods made from mixed mushrooms are strictly prohibited for sale on market. What about chemical poisoning? Chemical poisoning sometimes happens. It occurs, but it's not common. Usually it happens if somebody take some fruits and vegetables or berries after its treatment and sprayed with some pesticides, for example, organic insecticides containing lead or arsenic. If they made it by mistake, It's not common. Sometimes, in case of improper storage of food with high acidity, marinated or pickled vegetables in lead glazed pottery or in zinc container or cadmium lined containers because of chemical reaction of organic acid and heavy metal. Heavy metal may comes to food products and cause intoxication and can affect kidney, nervous system. It can be accumulated in bones in case of chronic exposure. Only specially certificated containers can be used for storage of food. How to avoid food poisoning with pesticides? In fact, a lot of pesticides used in agriculture. Usually they create it safe, but we need to follow its guide, its instruction, and we must wait enough time after pesticides used. And about this time usually is about up to 2 weeks. Usually pesticides affect nervous system. Sometimes pesticides can contaminate drinking water. And recently it was mentioned in use in some states in India. New unknown disease occurs, and expected it happens because of contamination of drinking water with some pesticides, because pesticides, usually phosphororganic pesticides, affect nervous system, affect synapse, and sometimes it can cause death. Its mortality is about 8%. Heavy metals, as mentioned briefly above, for example, mercury. Usually intoxication with mercury happens if we use mercury-containing pesticides. Mercury can be accumulated in fish. If it's accumulated in sea or in river on the bottom, and in food chain, it can be transmitted from river to fish to humans. And for example, mercury intoxication caused Minamata disease. Its name becomes its origin related to one district, Minamata district, or its name of river, Japanese river. Or Itai disease, intoxication with cadmium. Or if some persons by mistake used zinc bleached from galvanized pot because of chemical reaction with organic acid and zinc, such as pickled vegetables or when boiling some fruits. Because of chemical reaction, zinc from galvanized containers can transfer it to food and cause intoxication. So pay your attention. Finally, focus what you must do as a doctor in case of food poisoning. First of all, sure, you need to organize medical care. First of all, you must organize medical treatment. Second step, to prevent intake of suspicious food suggested or source of contamination by other sick persons. We need to inform health care authorities about food poisoning to organize its investigation. To make investigation, doctors need to collect biological materials, for example, vomit and fecal, blood, urine for laboratory investigation to find bacteria or the exotoxins. How to make a conclusion about its investigation? Firstly, we need to suggest what diagnosis can be. The second stage is laboratory analysis of taken biological materials taken from patients. We need to provide a blood analysis with this immunological investigation, with the serum diagnostics. In case of little cases, coroner must take inner organs of stomach, liver, intestine to check it in a laboratory to find the bacteria or toxins. Healthcare authorities should organize sanitary inspectors to check condition of food catering objects, and investigation, microbiological analysis, taking samples from food and from kitchen equipment should match with the bacteria or exotoxins found in the patient's analysis. And if we done all these steps before, we can describe the food poisoning mechanism. What we need to do? We need to put affected person in the hospital. If the source of food contamination is a sick or bacilli carrier person, we need to provide his or her isolation for proper sanation and treatment. We need to prevent growth of bacteria, provide this disinfection, and contaminated food should be destroyed. And finally, we need to write the protocol about food poisoning investigations, explaining everything details, what is the source of origin, what is the food, what is the mechanism of transfer, and some information about number of affected persons, about their treatment and outcome must be mentioned in this protocol. Thank you, my dear students. Thank you for attending all these lectures. That's it for actual semester. Stay healthy and take care of yourself and persons around you.